This method allows researchers to collect pollen from bumblebee colonies with minimal disturbance to the bees and low risk of bee stings to the researcher. Using this protocol, pollen can be collected from multiple colonies simultaneously, permitting researchers to use more colonies with less labor and expense than hand collecting pollen. This method will benefit research in pollination and bee health, pesticide science, and environmental contamination, and can be adapted to multiple species of bumblebees.
Begin by removing the support structures printed with the trap body and catch basin, including those of the sieve structure and the trap body. Clear any plastic strands crossing the raised edges of the pollen filter with a hand drill. Then use a box-cutting razor blade and sandpaper to even out any bumps or raised edges on the flat side of the filter. Gently push the plastic pollen filter through one side of the trap body. The filter will only fit in one way, as the left side of the trap has a larger opening to accommodate passage of the raised filter cones. If the side slits are too small for the pollen filter to slide through smoothly, use a razor to scrape enough plastic away from the slit in the trap body. Ensure that the filter fits securely in place with no more than a 2 mm gap between it and the trap body. Slide the raised edges on the bottom of the trap body into the groove on top of the catch basin. The catch basin should be positioned directly underneath the sieve region of the trap body. Cut or sand the plastic to allow smooth placement and removal of the catch basin. Placement of the catch basin will secure the pollen filter into place.